Hello friends, welcome back to our YouTube channel. I hope you all are doing great guys. So in this video we are going to learn about plant growth and development chapter. So in this chapter we will discuss about these topics. That firstly we will discuss about growth, then differentiation and redifferentiation. These process we already covered in anatomy chapter. So in this chapter, we just read it. Then development and then plant growth regulators or plant hormones. So let's start this chapter. You have already studied the organization of flowering plant in chapter 5, that is morphology of flowering plant. Have you ever thought about where and how the structure like roots, stems and leaves, flowers and fruits and seeds arise? So where the structure arise and how the structure arise on a plant, and that too in an orderly manner, orderly sequence also. Like firstly roots grow, then stem, then leaves, then flowers, then fruits and seeds. So it is also in an orderly manner. You are by now aware of the terms seed, seedling, plantlet and mature plant. You have also seen that trees continue to increase the height. So as we know, the tree continues to increase their height throughout their life or girth over a period of time. That is the width of the plant. Girth is the width of the plant. However, the leaves, flowers, and fruits of the same tree not only have limited dimensions but also appear and fall periodically. So, trees do not have limited dimension or shapes. It has structure like stems and leaves grow and periodically it falls. Why does vegetative phase precede flowering in plant? So why this vegetative phase not come before plant, before in flowering plant, earlier in flowering plant? All plants organs are made up of variety of tissues. All plants are made up of variety of tissues. Is there any relationship between the structure of cell, a tissue, an organ and the function they perform? Is there any relationship? Can the structure and function of these be altered? And the cell structure and this function, tissue and organ function, is being altered or being changed? All cells of the plant are descendants of the zygotes. See, all the structure of the plant are descendants of the zygote, or all the structure of plants are formed by the zygote. Zygote is the initial structure. The question is then why and how. This is important line, that all cells of a plant are descendants of the zygote. So the question is then why and how do they have different structural and functional attributes? So development is the sum of two processes, that is growth and differentiation. So development is the sum of two topics, that is two process, that is growth and differentiation. To begin with, it is essential and sufficient to know that the development of a mature plant from a zygote or fertilized egg follow a precise and highly ordered. So basically, development of plant is started with the zygote. Zygote is nothing but the fertilized egg. So then after formation of zygote, it follows a precise and highly ordered succession of events to grow the plant. During this process, a complex body organization is formed that produce. So during this process, this highly and precise process that produce roots, leaves, branches, flowers, fruits, and seeds. Eventually, they die, and eventually, periodically, they die. The first step in the process of plant growth is seed germination. So this is also important. So the first step in the process of plant growth is the seed germination. So firstly, seed germinate. The seed germinates when favorable condition for growth exists in the environment. When a favorable condition for seed like proper encourage or soil, uh, air, water, sunlight. So all these conditions are favorable for its growth. In absence of such favorable condition, the seeds do not germinate and goes into a period of suspend growth or rest. So, in absence of these condition or these favorable condition, seed undergoes dominance, dominance stage or the rest stage. Once favorable conditions return, the seeds resume metabolic activities and growth takes place. So, in this chapter, you shall also study some of the factors which govern and control these developmental process. These factors are both intrinsic or internal factors and extrinsic or external factor. Intrinsic internal factors like its hormone availability of water and all that, and extrinsic or external factor is that outside temperature and water, air. Now let's start with growth. Growth is regarded as one of the most fundamental and conspicuous characteristic of living being. What is growth? Growth can be defined as an irreversible, permanent increase in size of an organ. So, growth is a irreversible process. It is not reversed with time. At first, plant height is increased, then after some time, it decrease. So, no, growth is irreversible process. If it is increase, it always be increase. So, growth can be defined as an irreversible, permanent increase in size of an organ or its parts or even an individual cell. Generally, growth is accompanied by metabolic process, both anabolic and catabolic. So, growth is dependent on both type of process like anabolic process and catabolic process. And in anabolic process, the formation of substance takes place or the molecule takes place, and in catabolic process the breakdown of molecule takes place that occur at the expense of energy. So both these process require energy. Therefore, for example, expansion of leaf is growth. So leaf expansion is growth. How would you describe the swelling of piece of wood when placed in water? So when we place a piece of wood inside water, after some time it swell and become large in size. So is this process is a example of growth? So no, this process is not example of growth. It is a imbibition process. So it is occur by due to osmosis. Here germination and seedling development in a bean is shown. So this is soil line, this is seed and hypocotyl cotyledon. After roots arise, epicotyl portion outwards, these cotyledons, epicotyl and hypocotyl. Plant growth generally is indeterminate. So plant growth generally is indeterminate, which means plant keeps growing throughout their life. Plant keeps growing throughout their life. So that's why plant growth generally is indeterminate. Sometimes new leaves, roots, and stems form. So it is due to meristems of plant. So plant growth is unique because plants retain the capacity of unlimited growth throughout their life. This ability of the plant is due to presence of meristem at certain location in their body. So this ability is due to meristem. The cells of such meristems have the capacity to divide and self-perpetuate. The product, however, soon loses the capacity, and the product of these cells, meristem cell, soon loses to capacity to divide, and such cell make up the plant body, and that cell which loses the capacity to divide make up the plant body, and meristem further form the new plant bodies like new leaves, new roots, and new stems, etc. This form of growth, wherein new cells are always being added to the plant body by the activity of the meristem, is called the open form of growth. So that type of growth in which new cell is always added to the main plant body, that type of growth is open form of growth. What would happen if the meristem ceases to divide? So if meristem ceases to divide, then no organ, like no part of plant is grow, like no new leaf form, no roots form. Does this ever happen? So no. In earlier classes, So in earlier classes, you have studied about the root apical meristem and shoot apical meristem. So we studied these topics in anatomy. You know that they are responsible. So root apical meristem is located at the tip of the root, and shoot apical meristem is located at tip of the shoot. So you know that they are responsible for the primary growth of the plant and principally contribute to the elongation of the plant. So meristems, so root apical meristem or shoot apical meristem are for primary growth of the plant and for its elongation or increase in height along their axis. You also know that in dicotyledonous plant and gymnosperm, the lateral meristem, vascular cambium, and core cambium appear later in life in dicot plant and gymnosperm. So the lateral meristems, cylindrical meristem, vascular cambium, core cambium, these type of tissues appear later in life. These are the meristems that cause the increase in girth. So these are the secondary meristems which increase in girth or the increase width of the plant or its part in which they are active. This is also known as secondary growth of the plant, and this is secondary growth of the plant. Growth is measurable. Growth at cellular level is principally a So here diagram is shown that diagrammatic representation of location of root apical meristem and shoot apical meristem and vascular cambium arrows exhibit the direction of growth of cell and organ. So shoot apical meristem and root apical meristem increase length of that organ, and vascular cambium increase girth or the width of the organ. So growth at several level is principally a consequence of energy in the amount of protoplasm. So if we seen growth at cellular level, then we see its protoplasm level. Since increase in protoplasm is difficult to measure directly, one generally measures some quantity which is more or less proportional to it. Growth is therefore measured by a variety of parameters, some of which are increase in fresh weight, dry weight, length, area, volume, and cell number. You may find it amazing to know that one single maize root apical meristem can give rise to more than 17,500 new cells per hour. So this type we measure growth. So one single midge root apical meristem give rise to 17,500 new cells per hour, whereas cells in a watermelon may increase in size by up to 350,000 times. In the former, growth in the former or the number, that is one root apical meristem of maize is formed 17,500 new cell per hour. Is in this, growth is expressed as increase in cell number, and later in case of watermelon, growth as increase in size of the cell. While the growth of a pollen tube is measured in terms of its length, and growth of pollen tube here is measured in terms of its length, and increase in surface area denotes the growth in a dorsiventral leaf. So in dorsiventral leaf, if its surface area is increased, it shows the growth of leaf. Phase of growth. The period of growth is generally divided into three phases, namely meristematic, elongation, and maturation. So this topic we already discovered in anatomy of flowering plant, that meristematic and elongation and maturation. Let us understood this by looking at the root tips. So the constantly dividing cell both at the root apex and the shoot apex. So that cell which are constantly dividing at the apex of shoot and the apex of root represent the meristematic cell and meristematic phase of growth. The cell in this region are rich in protoplasm. So in this region, that region cell at tip of shoot and tip of root are rich in protoplasm, possesses a large conspicuous nuclei. Their cell walls are primary in nature, thin and cellulosic with abundant plasmodesmatal connection among each other. The cells proximal or just next away from the tip, or let's say this is root tip. Here root apical meristem present, and just above it, just proximal to it, or here meristematic zone represent the phase of elongation. Here cell elongate to increase length, increased vacuolation. So cell elongates by increased vacuolation. Cell enlargement and new cell wall deposition are the characteristic of the cells in this phase. Further away from the apex or above this portion, so further away from the apex, that is more proximal to the phase of elongation, lies the portion of axis which is undergoing the phase of maturation. The cells of this zone attain their maximal size in terms of wall thickening and protoplasmic modification. Most of the tissue and cell types, so this zone, so this is meristematic zone, this is elongation zone, and this is maturation zone. Now let's read growth rates. The increased growth per unit time in terms of in termed as growth rate. Thus, rate of growth can be expressed mathematically. An organism or a part of organism can produce more cell in a variety of ways. So, two types of that is arithmetic and geometric. So, in case of geometric growth, like this is one cell which has capability to divide, and this one cell form two new cell, and these two new cell also have capacity to divide. So each cell form two new cell, so it makes four new cell, and these four make cell 16 cell, and these 16 cell make 32 cells. So this is an example of geometric growth. This has happened here. Example: when zygote cell divided, then it forms 1 to 2 and 2 to 4. Here it is geometric phase. All cell divided in geometric phase. But in case of arithmetic phase, only one cell divide, rest cells are uh, differentiated. Only one cell is undifferentiated or divided, rest cells are differentiate or not divide. So one cell, only this one cell divide, that blue cells, dark blue cell divide, and rest cells are differentiate. So after zygote phase, there is a phase, arithmetic phase, in which only one cell has capacity to divide and rest are differentiate. So A arithmetic, B geometric growth है. Stages during embryo development, during geometric phase, and arithmetic phase after embryo development. The growth rates shows an increase that may be arithmetic or geometrical. In arithmetic growth, following mitotic cell division, only one daughter cell continues to divide while the other differentiates and matures. The simplest expression of arithmetic growth is exemplified by root elongating at a constant rate. So, arithmetic example is root elongating at a constant rate. On plotting the length of organ against time, a linear curve is obtained. Mathematically, it is expressed as LT equal to L naught plus RT. That is, LT is length at time t, L naught is length at time 0 or initial length, and R is growth rate or elongation per unit time. So, by y equal to mx plus c. C is L naught, that is intercept. So here this is initial height of the plant. L naught is initial height of the plant at time, let's say at t equal to 0 time, and the m is slope and x axis. So time is at x axis, and r, that is slope, R. Slope, that is growth rate or elongation per unit time. So as time increase, height of the plant is also increased. So this is example of arithmetic growth. Let us now see what happens in geometrical growth. In most systems, the initial growth is slow. So in most systems, the initial growth is slow because let's say example, initially one cell have capacity to divide, it makes two cell, then it makes four cell. So initially this type of growth is slow, and after some time, let's say at 16 cell condition, so 16 cell make 32 new cell. So after some time, growth rapidly increase. Initially it is slow. So in most system, the initial growth is slow, that is the lag phase, and it increases rapidly thereafter at an exponential rate, or that is log or exponential phase. Here, both the progeny cells, or the both the progeny cells, that is uh, these both progeny cells following mitotic cell division retain the ability and continue to do so. However, with limited nutrient supply, the growth slows down, leading to a stationary phase. So, at this time, Nutrient supply is limited. So due to nutrient supply, limitation of nutrient supply, stationary phase occurs. So initially lag phase, then exponential phase, then stationary phase. Stationary phase is due to limitation of nutrition. If we plot the parameter of growth against time, we get a typical sigmoid or the S curve. A sigmoid curve is a characteristic of living organism growing in a natural environment. It is typically for all cell, tissues and organ of a plant. Can you think of more similar example? What kind of a curve can you expect in a tree showing seasonal activities? The exponential growth can be expressed as, that is w equal to w naught e to the power rt. That is w1 is the final size, that is weight, final weight, final height or number, and w naught is initial size, height, number, and r is the growth rate, t is the time of growth, e is the base of natural logarithm. So here R is the relative growth rate and is also the measure of the ability of the plant to produce new plant material. So R is relative growth rate and it also the measure of ability of the plant to produce new plant material. So R is the measure of ability of the plant to produce new plant material, to form new plant material. So it is the ability, it is the measure of ability of the plant to form new plant material, and it is also relative growth, referred to as the efficiency index, and it also refers to as efficiency index. Hence the final size w1 depends on the initial size, that is w naught. So final size depends on initial size. So here comparison between absolute and relative growth. So absolute growth is total growth of a plant part in with per unit time, and relative growth is growth with initial parameters. So let's say here total growth is that is 5 to 10 centimeter. That is total growth of leaf is 5 centimeter square, and here 50 to 55, that is 5 centimeter square. So in both cases absolute growth is of 5 centimeter square. But if we look about that relative growth, so 5 to 10, that is 100% growth, and here not 100% growth. So relative growth is not same in both the leaves. So both leaves A and B have increased their area by 5 centimeter square in a given time to produce A1 and B1 leaves. Quantitative comparison between the growth of living system can also be made in two ways. That is, first measurement and comparison of total growth per unit time. So total growth per unit time is called the absolute growth, and the growth of the given system per unit time expressed on a common basis. Example, per unit initial parameters is called the relative growth. So total growth per unit time is absolute growth, and growth with initial parameters is relative growth. So two leaves A and B are drawn that are different in size but shows absolute increase in area in the given time to give leaves A1 and B1. However, one of them show much higher relative growth. That is, A leaves A leaf show much higher relative growth. So which one is where? That is leaf A. So let's read condition for growth. Why do you not try to write down what you think are necessary conditions for growth? This list may have water, oxygen, and nutrients are very essential elements for growth. The plant cell grows in size by cell enlargement, which in turn requires water. Turgidity of cells help in extension growth. So turgidity of cell help in growth extension. Thus, turgidity of cell, that is by inside a pressure to outside wall. So it extends the growth and extends cell. Thus, plant growth and further development is intimately linked to the water. So it is intimately linked to the water because due to water pressure exert on outward wall. Status of the plant. Water also provides the medium for enzymatic activities, and it also provides medium of enzymatic activities like cytoplasm needed for growth. Oxygen help in releasing metabolic energy essential for growth activities, that is for respiration. Nutrients, macro and macro essential elements are required by plant for the synthesis of protoplasm and act as source of energy. In addition, every plant organism has an optimum temperature range best suited for its growth. Any deviation from this range could be detrimental to its survival. Uh, uh, we have already seen biomolecular chapter. Environmental signals such as light and gravity also affect certain phases, stage or stages of growth. So environmental signals like light and gravity also affect growth. Now, next topic is differentiation, de-differentiation, and redifferentiation. So these topics we already cover in anatomy of flowering plant chapter. So, firstly, there is undifferentiated cell, which is the dividing cell. So, dividing cell example is primary meristem and intrafascicular cambium, not interfascicular cambium. It is intrafascicular cambium. Primary meristem example is root apical meristem and shoot apical meristem. After undifferentiated cell lose the ability of division, and process is, that is differentiation. That is first process is differentiation, and after this process it forms primary permanent tissues like primary xylem and primary phloem. And after it again gain ability to division. That process is de-differentiation, and from uh, secondary meristem like uh, secondary meristem or cylindrical meristem, that is vascular cambium, and it helps in expand the growth or the girth of the plant. Then it again loses the ability to divide, and process is redifferentiation, and form secondary permanent tissue like secondary xylem and secondary phloem. So the cells derived from root apical meristem and shoot apical meristem and cambium differentiate and mature to perform specific function. This act leading to maturation is termed as differentiation, and here formed is primary permanent tissue. During differentiation, cell undergoes few to major structural changes both in their cell walls and protoplasm. For example, to form a tertiary element, the cells would lose their protoplasm because it forms primary xylem and primary phloem, which is a tertiary element. So, from tertiary element, it forms protoplasm. It loses their protoplasm. They also develop a very strong elastic lignocellulosic secondary cell walls to carry water to long distance even under extreme tension. Try to correlate the various anatomical features you encounter in plants to the function they perform. So plants show another interesting phenomenon, the living differentiated cells that by now host have lost the capacity to divide can regain the capacity of division under certain condition, and this phenomenon is termed as de-differentiation or the breakdown of the process, that is differentiation. For example, formation of a meristem or the formation of secondary meristem like interfascicular cambium core cambium from fully differentiated parenchyma cells. While doing so, such meristem or tissues are able to divide and produce cells that once again lose the capacity to divide but mature to perform a specific function. That this process is redifferentiation. List some of the tissue in a woody dicot redifferentiation process from that is dicot plant, that is secondary xylem and secondary phloem. There are the product of redifferentiation, that is secondary xylem and secondary phloem. How would you describe a tumor? What would you call the parenchyma cell that are made to divide under controlled laboratory condition during plant tissue culture? So tumor is a condition in which cell loses its ability to stop division. So in a tumor cell actively dividing cell and replicate again and again. Recall in section, we have mentioned that the growth in plant is open, that is, growth is indeterminate. New cell are always added to the main plant body. It can be indeterminate or determinate. Now, we may say that even differentiation in plants is open because cells tissues arising out of the same meristem have different structures at maturity. So, all these structures are different at maturity stage. The final structure at maturity of a cell tissue is also determined by the location of the cell within. For example, cells positioned away from root apical meristem differentiate as root cap cells. So away, that is here from root cap, and here is root apical meristem. So it is root cap. So away from meristem cells differentiate itself to form root cap for protection of these meristematic cells, while those pushed to periphery mature as epidermis, and those cells are towards periphery of the plant part is formed epidermis. Can you add a few more example of open differentiation correlating the position of a cell to its position in an organ? So with this we end this lecture. In part 2 of this chapter we continue this chapter with development. I hope you understand this topics very well. If you have any doubt, feel free to ask in the comment box. I'll definitely answer your doubt. Thank you very much. Bye bye.